let us talk about the base excision repair pathway. So the first step in this pathway is carried out by DNA glycosylase. This DNA glycosylase catalyzes the base excision to produce an AP site, which is apurinic or apyrimidinic site, because this enzyme removes the base from the DNA helix and generates an AP site. The next enzyme in the pathway is AP endonuclease, and this enzyme hydrolyzes the phosphodiester bond 5' to the AP site to generate a nick. So this second enzyme, this cuts the helix, cuts the strand from 5' to the AP site to generate a nick or a cut. So here you can see the first reaction caused the removal of a nitrogen base from here, and this created an AP site, and the AP endonuclease then cuts this from this 5'. So when this bond has been cut, a nick is introduced in the DNA strand. So this nick is introduced by AP endonucleases. So this is second step in the pathway. E. coli has two well characterized AP endonucleases, and these are exonuclease 3 and endonuclease 4. Exonuclease 3, despite its name, accounts for most of the bacterial AP endonuclease activity. Although its name is exonuclease, but it performs most of the endonuclease activity. Both are multifunctional enzymes that have 3' phosphate and 3' repair phosphodiester activities. So these enzymes can perform more than one activities. So they are called multifunctional enzymes. The former activity, that is 3' phosphate activity, removes phosphate group from the 3' end of a DNA strand. So from the 3' end, a phosphate group is removed by the action of these enzymes. The later activity, that is 3' repair phosphodiesterase activity, removes the 3' unsaturated aldehydic group that is produced by the DNA glycosylase lyase activity. After this activity, an aldehyde group is produced at this site. These activities are important because DNA polymerase cannot attach new nucleotides to a blocked 3' end, because this aldehydic group, it blocks the 3' end, and if this aldehydic group is present, new nucleotide cannot be attached here by the DNA polymerases. So the removal of this aldehydic group is essential. The mammalian AP endonuclease, which is APE1, is homologous to E. coli exonuclease 3.